Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Welcome to this bonus episode of the Drug Discovery World podcast. Today's episode is taken from one of our DDW webinars. On a side note here, apologies the sound quality isn't as good as usual, but if you enjoy this episode and it proves useful and popular, then we'll feature more episodes based on webinars for you in the future. If you'd like to view the original webinar recording with all the slides, then there's a link to the full video in the show notes. And if you enjoy this webinar episode, then you can find more webinars on the webinar section of our website. So as always, if you enjoy the podcast, then please do subscribe and leave us a review, and I hope you enjoy this bonus webinar DDW podcast episode.
Welcome everyone to this Drug Discovery World webinar entitled Best Practice Approaches and Overcoming Potential Challenges in Targeting Kinases for Drug Discovery. I'm Robert Jordan, Publisher and Editor-in-Chief of Drug Discovery World, DDW, and I'm going to serve as your moderator this afternoon. As we well know, kinases are one of the most widely studied drug target classes with a history of over 20 years in the drug discovery research spotlight. Indeed, the latest figure on the commercial impact of total sales of kinase-related drugs is estimated at $240 billion during the period between 2011 and 2015. Which naturally supports the continuation of targeting kinases for the development of new drugs, not only in cancer, but also for many other indications. With that in mind, selecting optimized assays for screening, hit validation, selectivity profiling, and lead optimization represents key steps towards identifying candidate compounds for preclinical and clinical development. But with over 500 related proteins in the kinome, the path to candidate selection is not always straightforward. So today's webinar will discuss the need for both targeted activity as well as appropriate selectivity as key requisites in the discovery of new kinase therapeutics. Ultimately, this should result in a kinase inhibitor for therapeutic application that demonstrates specific activity at the intended target while also being selective enough that its therapeutic goal is achieved without causing unmanageable toxicity. So I'm delighted to be joined today by Dr. Alistair King. Alistair has over 17 years of research experience with a focus on oncology drug discovery, cellular assays, and laboratory automation. And he's had increasing responsibilities at SmithKline Beecham, as it was, latterly, obviously, GlaxoSmithKline, Johnson & Johnson, and more latterly with today's sponsors, Eurofins Pharma Discovery Services, where he is group leader of the OncoPanel cell-based profiling service. So, if you're ready, Alistair, the floor is indeed yours.
Well, thank you, Robert. Thank you for that kind introduction. It's a pleasure to be here. Hello, everyone. I'm looking forward to talking with you today about various aspects of targeting kinases in drug discovery. We're going to cover various aspects of kinase drug discovery, but first of all, I'd like to just give you a little bit of background as to where I work. I work with Eurofins Pharma Discovery Services in St. Charles, Missouri, in the United States. We are a pharma services CRO company, and we offer a range of services, including what used to be the former Upstate Kinase Panel. Many of you will be familiar with that. Those of you that aren't, it was recognized as being a gold standard in the industry for kinase work. So what we're going to cover today is the whole range of kinase drug discovery, talking to start with a little bit about kinases in drug discovery, how they came into being and some of the successes, and then we'll get into the actual process of conducting kinase drug discovery, starting in a logical sense with screening, then going through hit confirmation, taking a little bit of a look at mechanism of action and why one might need to evaluate that, and then going through to selectivity profiling, the importance of getting selectivity against the kinase of interest, and then moving on into some of the other studies necessary for kinase drug discovery, including cellular studies, and then beginning to wrap up towards the end, looking at other target classes that may be relevant to kinase drug discovery, and other aspects of a drug discovery program that, while not kinase-centric, are still necessary for a successful kinase drug discovery program. So that's the agenda that we will be covering. What we will start with here is an overview of kinases in drug discovery. So the history goes back several years now. Sarc was first identified as a human oncogene, the first oncogenic protein kinase, back in 1978. That really set in path the motion for a new target class that potentially could become druggable targets. Now, it was interesting because all kinases employ ATP as the phosphate donor molecule. And so there was some skepticism in the field as to whether one could actually develop selective kinase inhibitors, given that ATP is required for the activity of all kinases. So there was a little bit of concern as to whether this would in fact be possible. Looking at 1991, sporosporin was first identified and classified as a nanomolar inhibitor of protein kinase Cs. And then going through work that was done subsequent to that, the first kinase structure was identified also in 1991, that being for protein kinase A. And then looking forward to that and sort of cutting through the timeline a little bit, the Upstate panel was launched in 1999, and the bottom line is that the kinases were eventually found to be druggable targets. From a personal perspective, I do remember being at the first rollout of the clinical data for Gleevec many years ago now, but it's shown that kinases in fact can be druggable. And it is possible to get appropriate levels of selectivity depending upon the indication that is required for the specific drug discovery program in question. So along that line then, there have been numerous kinase inhibitor drugs developed. Drug discovery really is nothing new. It's something that the humans have actually taken advantage of since days of antiquity. Early medicinal uses of things like honey as anti-infective agents and things like that were known back in Egyptian times. But perhaps in more recent years, there are compounds that have been used for various human ailments, going from digitoxin that comes from the foxglove plant for treating various coronary issues, right the way through to aspirin from willow in the 1890s, to penicillin in 1928 as an antibiotic, conotoxin from the cone snail, which is used to treat disorders associated with voltage-gated calcium channels, and then one that's close to my background, being an oncology drug discoverer, paclitaxel, or Taxol, which has been extracted from the yew tree, which is used as a treatment for various cancers, in particular breast cancer. Now, what is striking about this is these are all natural products, but they are actually, the natural product pool has actually been the most successful source of drug leads. Now, what should be taken away from this is that these drugs have been developed by the various organisms from which they were identified over the course of thousands, if not millions of years, to fight off predators and so forth. Now, we have to undertake drug discovery, obviously, in a much shorter time frame, and we have to develop the same degree of efficacy of compounds and safety and so forth. So just walking you through the process, targets are traditionally validated, and that might be by genetic means or a disease association of a mutation. There is a link between a given target and a disease state. And then that target is developed into an assay which is used for high-throughput screening purposes. The high-throughput screening I always liken to looking for a needle in a haystack. Following high-throughput screening, then leads or early hits are actually taken from that, validated as hits, and developed through a process of lead optimization and through the preclinical development stage, ultimately to where what's called an IND, or investigational or new drug application, is made, and then that compound is taken onwards into the phase 1, 2, 3, and 4 clinical trials. Now, what's important to remember here from the drug discovery process is that once a hit is identified as being an inhibitor, or in some cases an activator, of a given target in a high-throughput screen, various parameters need to be satisfied in order to develop that hit into a drug. The hit must have, and the lead that is developed from it must have appropriate activity at the target. It must have potency, and that potency can be dependent ultimately upon the disease indication and other factors, including some of those that I'll mention in a second. The hit must, or the lead and ultimately the drug, must have appropriate selectivity, and that selectivity, again, to some degree, is dependent upon the indication for which the drug is being developed. Going forward then, translating activity, potency, and to a degree selectivity, that goes into efficacy, which traditionally is in vivo models. We will talk a little bit about cellular efficacy later on in this presentation, and that is a step at which the activity of the drug, especially in the in vivo models, is shown to work in a living organism, traditionally small rodent models to start with, but then more advanced species. Coupled with that is the evaluation of bioavailability, and bioavailability simply is a measure of how much of that drug is being taken to the target in the system. Obviously, safety is a key part of the development of the drug there as well. You've heard me mention the concept of a high-throughput screen, which has been the mainstay of screening for not only kinases, but a wide range of different drug targets for many years. High-throughput screening typically evaluates of the order of hundreds of thousands to millions of compounds. It comes with various advantages, including the fact that hits identified from high-throughput screening generally are likely to give higher diversity because of the broadness of the chemical pool from which the high-throughput screen is conducted. With that comes the potential for novel IP, also the possibility for novel mechanisms of action. Now that can be as a result of the chemical series that are identified and also the assay that's used. But there is also the possibility to pull out relatively high throughput hits from a high throughput screen, while traditionally they're not usually at the sufficient activity required for development into a drug. It is still nevertheless possible to pull out higher affinity hits. And you'll see why I mention that in a second. There are, of course, some disadvantages of a high-throughput screen because of the broadness of the compounds there. There is the potential for off-target effects, especially if the high-throughput screen compound collection contains compounds that are known hits of other target classes. It may be that they actually form great kinase inhibitor drugs as an early hit, but because of their background, then they do have activities that would need possibly to be dialed out from their profile to make that compound into a drug. One of the potential cons is tractability. One doesn't know necessarily whether a hit coming out of high throughput screening will be tractable. There are obviously other things that need to be done to develop a hit into something of adequate potency and selectivity, bioavailability, and so forth. And that's really where the power of medicinal chemistry comes into being in resolving some of those issues. Obviously, high-throughput screening is a fairly time-consumptive and relatively costly procedure. It takes a while to run a high-throughput screen. And with that is also the need for the presence of relatively high-cost instrumentation to conduct high-throughput and ultra-high-throughput screening. Fully automated robotic systems are required, and obviously that it requires investment that perhaps smaller companies and academic institutions may not necessarily have access to. That doesn't mean to say they can't do that, but it is still a consideration. Then moving forward into focused sets, that has been a popular method of screening where one is looking at sets of compounds that are more focused in either their activity or their history within a certain area. An example here would be a kinase-focused set, and numerous companies have put together panels of inhibitors that have come from previous kinase drug discovery programs or frameworks that have been taken from the literature. Those compound selections are obviously a little bit smaller, of the order of thousands to tens of thousands of compounds. What is attractive about those is that they provide the potential for similar mechanisms of action, and because a lot more is known about those compounds, there is the potential for better tractability of those hits. They obviously have known behavior, and because the screening there is a little bit smaller than a high throughput or an ultra high throughput screen, the cost and time factors are more advantageous. So, of course, there are some cons. One of the issues is the off-target effects within the target class. If you're starting with an inhibitor that was developed towards kinase X and you're looking at kinase Y, it may be that you do not want kinase X activity in your ultimate profile, so that would have to be dealt with, again, depending upon the indication. There is also the potential for narrower IP space because a lot of those frameworks will likely have already been patented, especially if they are representative of late-stage lead compounds. There is also the potential for less novelty, again, because more is known about the compounds in that collection. But it does provide a very quick and relatively easy way to get a hit, and is particularly useful for identification of tool compounds, which may feed back into target validation studies. Then just looking at reduced complexity screening or fragment-based screening, this is a method whereby smaller molecules, and of the order of hundreds to a couple of thousand molecules, are typically looked at for binding to specific areas of a kinase based on binding energy. Now, the issue here is because their contact area with the kinase is smaller, their binding energy is lower, so their binding affinity is lower, so that is a negative. But because they're not larger molecules, and generally they satisfy the Lipinski rules amply, their efficiency of binding is a lot higher. Coupled with that, because they are not so large molecules, is that there is a much lower possibility of steric blocking of those smaller components, so they can get into the active site of the kinase or the area of the kinase that is being targeted and bind without another part of the molecule actually precluding that. One other thing is that it is possible to pull out more hydrophobic hits using reduced complexity screening, which is not always available from some of the higher throughput methods. So simply taken, then, a number of hits towards different components, different parts of the active site of the kinase or the site being targeted are then strung together, again, with the help of a medicinal chemist, and that provides a compound ultimately that has a higher binding and affinity and really is effectively a designer compound. One of the cons of this is that, again, there is the need for high-cost instrumentation, typically more analytical, chemical-based, mass spec, NMR, things like that. And there is also the need for crystallography, so coming with that often larger amounts of protein to be purified to high degree such that the synthesized molecules can then be co-crystallized with the kinase to make sure that the binding that is predicted has actually been modeled appropriately and that the final compound is really what you have actually set out to accomplish. So following in the wake of a high throughput screen then, a number of hits are identified. One of the first things really is the deconvolution of those hits and validation that the hits are in fact true hits. Traditionally, this is often done using the assay, and I'm not really going to go into specific assays. There are many different formats of assays that are appropriate for kinase drug discovery, ranging from simple fluorescent polarization binding assays, whereby a fluorescently labeled probe is used in a binding displacement assay, right the way through to more elegant studies, including some of the TR-FRET and HTRF-based assays, whereby a substrate, typically a peptide or a protein, is phosphorylated, and that phosphorylation event is detected immunologically. One of the key aspects for a high throughput screen, of course, is that the assay must have appropriate sensitivity, the ability to be miniaturized, and a suitably low cost enabled to be able to run the screen. Obviously, for more focus set screens where the size of the screen is smaller, the cost issue for the assay may be sacrificed for the advantage of other aspects of the assay that best fit your needs. One of the other things for deconvolution of a hit, in addition to identifying and validating that the hit truly is a hit with the assay, so that's a repeat of the measurement of activity of that hit, is the use of an orthogonal assay. And I'm just showing an example here that we at Eurofins have, whereby we have a TR FRET-based platform wherein we can measure single point confirmation or IC50 data in a medium to high throughput screening format. We can screen significant amounts of compounds and generating large amounts of data, typically for the numbers of hits that one would get for a high throughput screen. The assay is robust with a suitably high Z prime value and a low signal to noise. The next point about hit validation is it is one thing to identify a hit if you're screening in single point at something like 10 micromolar. That hit may be reflective of something with an IC50 perhaps in, say, the 3 micromolar region, or it may be a hit that has a 1 nanomolar IC50. So it's very important with your hits coming from a high throughput screen, once they have been validated, to confirm their activity and to measure their IC50 values. So it is important to take your hits, to validate them, and then to confirm their activity by IC50 confirmation. That really sets the stage for what work you have to do in terms of potency. If, for example, you do pull out hits from a high throughput screen wherein the activity of the compounds shows nanomolar potency, that may be getting very close to what you need for your particular indication. Alternatively, you may be pulling out hits with micromolar activity that look particularly desirable from an IP, chemical IP perspective or tractability perspective, and there is some work needed there to actually dial in greater potency. And again, that's where the power of medicinal chemistry comes to bear in being able to optimize those hits. So once one's done screening and confirmed the hits, what next? What really needs to be done to understand those hits? In particular, I'm going to move now to mechanism of action studies. And it is important to understand how your compound is working at the kinase target in question. Compounds obviously have more characteristics than just inhibition potency, and there are a range of enzymological assay formats and ways of doing enzymatic assays for kinases that can be used to identify if a hit, if for example it's an ATP competitive hit versus a non-competitive hit. So I'm just giving a summary here of why mechanism of action is important. It is important to understand additional characteristics of the inhibitor that you have. Residence time at the target can impact the pharmacodynamic activity of the compound. Giving two examples, fatinib is an approved oncology agent. It's actually an irreversible inhibitor, so that may have consequences for how you need to dose or the effect on the target. And again, that to some large degree is dependent upon the indication for which the kinase inhibitor is being developed. Lapatinib is another good example. It has an incredibly slow off rate, so that actually contributes significantly to its very good activity, and it's an approved agent now for the treatment of breast cancer. There are obviously, as I alluded to previously, non-ATP site targets that can have or can manifest themselves in improved specificity. If a compound binds to, in this case I'm using the example of sirolimus, which is a rapalog that can bind to the FKBP12 protein, that can actually modulate kinase activity in a mechanism that is not ATP competitive. There is also the potential, of course, to be able to develop inhibitors that bind elsewhere to the kinase that will regulate or modify its structure, which ultimately will affect its catalytic activity. but are not ATP competitive. And again, knowing this and knowing what you're trying to develop in your kinase inhibitor drug is important because the two are intimately tied. Lastly, there are, of course, in several disease areas, and in particular in the oncology field, numerous examples of kinases that develop mutations, often as a mechanism of resistance towards kinase inhibitors, but also as somatic mutations and so forth, which give rise to oncogenes in their natural environment. The two examples I've given there, osimertinib, targets mutant forms of the EGF receptor, which often are developed, in particular the T790M mutation, when cancers that are driven by EGF receptor signaling are treated by EGF receptor kinase inhibitors. And then lastly, a compound close to my heart, because I worked on it in a former life, dabrafenib, is an example of a targeted agent which specifically inhibits a mutant form of the BRAF enzyme, which is prevalent in a number of human cancers, and in particular melanoma. And that's a good example where a kinase inhibitor was developed towards a disease-associated mutation. Now, why is it important to perform kinase profiling? We've already established that we can identify and develop hits that have appropriate potency, but selectivity is an important aspect now. It is important to identify information about compound selectivity, and prediction of off-target effects has often what can be very severe impacts on safety. If one is developing a kinase inhibitor, there are certain kinases that one would not necessarily want to inhibit, and if that is part of the profile of a compound going forward, it may lead to unwanted or unacceptable safety profiles in vivo models. That is obviously something that, again, a medicinal chemist can address and can rectify. Certain off-target activities can be dialed out by tweaking the molecules appropriately while still maintaining activity and potency at the desired target. The other thing with profiling is it does give some insights into the effect on downstream proteins, and in particular cellular phenotypes. As I'm sure everybody is aware, the older idea of kinases being part of linear signal transduction processes is dated. And it's now known that just about every kinase is involved in a network of signaling events that is really very complex. So if you have other off-target activities, they may be deleterious to the ultimate effect of your compound inhibitor, or they may actually contribute to it. And again, that is dependent upon the indication for which your kinase inhibitor is being developed. Updating a profile is another important aspect. In some cases, there are kinase inhibitors that have been developed a few years ago when kinase panels were a lot more limited in size. And identifying additional targets may explain either why a safety issue was encountered with those kinase inhibitors, or looking at it from a more positive perspective, may actually expand the indication of existing assets. And that's an important aspect of looking at existing medications, which one already knows are safe, but then providing medications then that can treat other diseases and ultimately helping patients that are in need. So then moving forward again with the whole idea of looking at selectivity, where do you start out? There are upwards of 500 kinases in the human kinome. Which ones do you really want to look at? And in large part, that depends upon, again, the role of the kinase inhibitor for which you're designing a drug. One might want a very selective profile, and this is typically seen with a lot of drugs for things like inflammatory indications. The rheumatoid arthritis example is a good one. Whereas for the oncology space, the hurdle's a little bit lower, and there is generally the acceptance that while it is a good idea to identify targeted, very selective agents because they lend themselves to the potential for better combination studies, there have been numerous anti-oncology drugs that have been developed that have what one might call a dirtier profile. Smaller subsets can definitely provide informative starting points, but periodically checking for a more diverse panel is often a good idea because there may be other activities that either come and go as the medicinal chemistry campaign develops. Looking forward then at a case study, this is just an example of compound selectivity across the kinome. We looked here at 158 diverse small molecule kinase inhibitors across 234 human wild-type protein kinases. And there is a different spectrum of activity seen. And I'm not really going to go into detail with any one of these, but there are some key findings from this. One of the benefits of screening against a broader panel is that kinase inhibitors can then be repurposed and new uses can be found for them. We'd also mentioned looking at previously unknown off-target effects. And perhaps most importantly, and this drives all medicinal chemistry campaigns for kinases, is the relationship between chemical structure and activity profile, or what is known as structure-activity relationship, or SAR, can be derived, not only for the primary kinase of interest, but also for the off-target activities. So then looking at what we might call kinases that are more promiscuous, if one looks at kinases versus a variety of compounds to the screen, there are some kinases that time and again do seem to show activity against inhibitors. Those kinases that are more promiscuous than perhaps others might also form the nucleus of, if not a selectivity panel, to at least raise awareness that if one sees activity against those, it's not an uncommon thing. Again, providing more knowledge on the kinase inhibitor being developed. Looking now at the promiscuous kinases, the distribution of these over the kinome, what is notable here is that many of them do actually occur in the tyrosine kinase section. So that's just something to be aware of. Unfortunately, within some of the kinases, the tyrosine kinases, there are examples that would not want to be part of a profile, as they would lead to safety issues. So with that taken together, one is beginning to now flesh out more information about the kinase, what its potency, activity, selectivity are. But really the next frontier to cross is the cell membrane. It is important that all kinase inhibitors being developed can actually cross the cell membrane. It's one thing to actually have an inhibitor that can inhibit a biochemical assay, but then when faced with the cell, if it turns out that kinase inhibitor is actually not penetrant to the cell membrane, the internal process whereby that kinase is normally active, then it's going to be unaffected by the inhibitor. So the bottom line is then that one needs to be able to dial in cellular permeability. This may be existent in compounds coming from a high throughput screen, or it may be something that has to be improved and developed. And the way to test that is by setting up a cellular mechanistic assay. This is effectively a cellular version of the enzyme assay in the kinase's natural environment. So this is obviously a lot more complex than what we can establish in a simple biochemical assay. It contains all of the other proteins present in the cell that can convolute and complicate the activity of that kinase, in addition to other signaling mechanisms that can also influence its activity. Now, cellular assays for kinases, and really for other targets, intracellular targets at least, are not always popular. Not to say that they're not executed, but they're not always popular for the very reason that you might actually lose or not identify inhibitors that actually have good activity at the target and for which cellular permeability can be dialed into the profile. So really there are two key aspects of a cellular mechanistic assay that are needed for effective drug discovery. One is that the throughput must be suitable for the program needs, and again that depends on the kind of throughput that you're looking at. The other thing is that if the throughput is high enough, there is the need to automate the process to enhance that throughput. In some cell types, the inhibitor may not necessarily behave the way you think. So a key take-home from this is that the biological complexity of the cell can sometimes give unusual results or slightly different results to one particular cell model. So I can't stress enough that where possible, it is important to conduct cellular mechanistic assays in more than one cell line. And the reason for that is sometimes the biology is a little different. sometimes the other components of the cell or other mutations, especially in a cancer cell, can result in very slightly different behavior of that compound. So it's important to get a feeling for that. Moving on from the cellular mechanistic assay then, there needs to be a functional context. And ultimately, this is getting towards the treatment of the disease state. Now, I'm not going to go into every kind of functional assay out there. These are the contextual assays that show an effect on the cell. They can range from inhibition of cytokine secretion by immunological cells through to what I'm going to talk about here, which is the inhibition of proliferation of human tumor cells. Now, Eurofins offers what we call OncoPanel, which is a cell-based profiling service that covers actually now over 300 diverse genomically characterized human tumor cell lines that span a broad range of different human tissue types and tumor types. Now, we can use these and profile inhibitors against these cell lines to show their activity profile. To summarize, we've looked at different approaches for profiling kinase inhibitors in the wake of a high-throughput screen. We've covered the basic aspects of screening. Obviously, that one can go much deeper into that. We've looked at achieving potency. We've looked at achieving selectivity, and I've used the Eurofins Kinase Profiler platform as a good example of addressing that. We've looked at the need for showing the mechanistic activity of a kinase inhibitor in a cell, which gets more towards the disease context of the model being used. And then the functional assay, and again, I've highlighted that using the OncoPanel cell-based profiling service that we offer with Eurofins. So collectively, all of these approaches here are designed to translate your activity from an in vitro enzyme profile into a cellular context to fully understand what your kinase inhibitor is doing, ultimately to treat the disease for which it's being developed, but also in a safe manner, and in some cases providing greater utility or repurposing for existing assets. But what I should add here, and we're not going to go into this today because this is beyond the scope of really what today is an in vitro talk, is this helps guide in vivo model choice, which is really when you get to looking at the model of disease in a living animal. So I just want to sort of begin to wrap up here. Other target classes can be important. Just because an inhibitor inhibits a kinase doesn't mean to say that it won't inhibit other target classes. And I'm just using GPCRs, ion channels, and phosphatases here as examples. So one has to always be cognizant of the fact that there may be other target types that are inhibited. And that really comes into safety profiling of a kinase inhibitor. So with that, I'd like to thank you all very much for your attention. I've certainly enjoyed walking you through some of the aspects of kinase drug discovery. And with that, I'd like to thank you for your time, and we'd be happy to field any questions.
Thank you, Alistair. Very interesting presentation, and I'm sure we'll have some questions on that. Indeed. Tell me, do you think that knockout studies, for example, using shRNA, have value when studying kinases for drug discovery?
Yes, they do have some value. One of the issues with shRNA is that, of course, you're not inhibiting the target in the same way that you would with a small molecule kinase inhibitor. So one of the caveats for that is you will lose interactions with other scaffolding proteins, other interacting substrates, or even other kinases that may be involved as part of a complex. So there certainly is value. It does provide potentially interesting results that can be used, but it should be taken with caution. Perhaps the better approach, and again, it depends upon the resources available, is to try and develop a tool inhibitor that doesn't necessarily have to be particularly selective, but at least potent enough to show an effect at the target. That, in some ways, then would give you a validation that the small molecule approach would work. And then, obviously, if that is a path down which you would want to go, then you could launch a full-scale drug discovery effort along those lines. I was actually involved in a program many years ago in a different life, where we were looking at several kinases agnostically, and the approach was taken to look at not only the biological tractability of the target by conventional target validation type studies, but also the chemical tractability. And so the ability to actually generate a tool compound fairly quickly and easily, we used as a measure of how potentially chemically tractable that target may be. So it all factors into the equation.
Excellent. I have a question here from, I'm going to try and pronounce this properly, Alejandro Marino, Chief Scientific Officer at Persa Medica. And his question is, how about inactive versus activated kinases?
That's actually a very good question. What I'm not sure if the question is asking is about looking at the class 1 versus class 2 kinase inhibitors from the perspective of in vitro assays, or whether it's looking at that in a more general sense. Obviously, there are different ways of inhibiting a kinase. You can inhibit it in the active-like conformation, where there is not turnover of substrate, but the kinase is still in an active-like state. So conformationally, it looks like an active kinase. That's often been seen with the RAF inhibitors, BRAF inhibitors, things like that, where although locked in an active conformation, they can then partner with CRAF and for other kinases, partner with other kinases that are not inhibited and thus signal. However, if you are targeting a kinase to inhibit it in the inactive-like conformation, then the potential for interaction and thus transactivating other kinases by a structural event, that is then not part of the profile of that compound. In terms of assays, one would have to think very carefully about how one would measure that. If you're looking for an inhibitor that locks in the inactive-like conformation, really the only way that you can do that would be ultimately by looking at a crystallography type effect, where you're actually looking at the structure. Certainly for ATP competitive inhibitors, things like that, you might be looking for the movement of the active site loop in or out of the way of the active site as a gauge of active-like or inactive-like conformation. I say active-like and inactive-like because in a lot of cases, there are kinases that are mutated, so they're not conventionally activated, but they are active-like because often the phosphorylation event mimics. Often the mutation, the acidic mutation, will mimic phosphorylation of key residues that are needed in part of the activation process. So really all I would say about that is that one would need to examine and carefully choose the assays to measure that and be cognizant of what you're actually doing biologically to the kinase. Of course, there are other ways to do it than just crystallography. One could look at binding studies if there is a binding partner and measure binding to another partner kinase.
I have another question for you, and this is from Michael Vogel-Gasang from Washington University School of Medicine. His question is, what do you think about the value of cell-based assays using phosphokinase antibody arrays?
Yeah, that is obviously an interesting area. The whole world has been revolutionized with the phospho-specific antibodies and things like that. I think there is value in that. That may well be the next step that one could go to. If I'm understanding the question correctly, or at least the sentiment of the question, the potential applicability of that for looking at selectivity in a cellular context. The power of that is potentially tremendous, because that would then be looking at, in effect, the kinds of selectivity that we've been discussing in the webinar, but from a cellular perspective, where sometimes things can be a little different.
Okay, I think we've got time for one last question. This is from Isaac Johnson from Cancer Research UK. His question is, multiplexing is now quite common in shRNA screening and reduces costs and time. Do you think there will ever be something similar for in vitro assays?
That is a good question. I don't know. I think certainly from the cellular perspective, there is the potential to do that. I mean, what we do with many other assays, I mean, we talked about the OncoPanel platform there, where we will multiplex different readouts to get more information about the activity of, a kinase inhibitor, or in that case, other classes of inhibitors. The assay is not solely limited to kinases. In terms of comparing that with shRNA screening, I think there is potential for that. I think it's emerging at this point. So I think time will tell as to how beneficial that will actually be. But I think there is potential there, certainly in terms of being able to multiplex different approaches and to examine things concurrently. One of the key things there is to be able to look at all of the relevant pathways that are associated either directly with the inhibition, or coming back to the previous question about the cell-based assays with the phosphokinase-specific antibodies and things like that, being able to see just how the interactions between pathways play out. Certainly, as we discussed in one of the earlier questions, I'm a big proponent of going for an approach that is more akin to what one would be doing in terms of the therapeutic intervention, i.e. using tool molecules. While I understand and respect the value of shRNA studies, I'm always skeptical of their true value, because you're actually doing something very different to the protein than you would therapeutically.
Excellent. It actually looks like we've run out of time now. So once again, a big thanks to our speaker, Dr. Alistair King, and thank you to our sponsors, Eurofins Pharma Discovery Services. Thank you very much, and goodbye.
Thank you all.